the second short talk of today, which is by Fabian Müller, who is a junior professor at Saarbrücken University, leading his own group on integrative cellular biology and bioinformatics. And he will talk today about chromatin and gene regulatory dynamics of the developing human cerebral cortex at single cell resolution. Perfect. We can perfectly see your screen. And Fabian, the floor is yours.
Thank you. And thanks so much for putting together this awesome meeting. Thanks, Jörn, Maria, and Nina. I don't know how you managed to get this perfect lineup of speakers always. It's really a great meeting. I'm having a good time. And also thank you for giving me the opportunity to share some of my recent research here, where we looked at the chromatin dynamics in single cells as the human brain develops. And this is joint work together with Alex Trevino, Femina Anderson, and Lakshman Sundaram from the Pascal and Greening labs at Stanford. So as you all know, the human brain is a highly complex organ, and we have many very important developmental processes going on, particularly early on in embryogenesis. For instance, neurogenesis is happening in weeks 8 to 20, 24 something in the developing embryo, which really gives rise to a complex tissue structure that ends up in this layering pattern here, where we have different neurons occupying different layers, and we have radial glia cells, which really give rise to this to those neurons through the emergence of also intermediate progenitor cells. And we looked at this now from using single cell RNA sequencing and single cell attack sequencing from primary human samples that we were fortunate enough to get through the collaboration with Cedric Pester's lab at mid-gestation, so ranging from week 16 to 24 after conception. So this is how the corresponding UMAPs look like. On the left-hand side, you can see the single cell RNA world, and on the right-hand side, you can see the single cell attack world. And of course, we did what most people do. We did some just dimensionality reduction, clustering, and what you can already see is that the two landscapes actually correspond quite nicely to one another. So we have sort of a stem-like structure here, which is occupied by the radial glia cells. We have a potential differentiation towards the oligodendrocyte progenitor cells here. We have this large branching out towards the excitatory neurons, and we have sort of the outgroups here by the inhibitory neurons. And the same is true for actually the attack side of things. What we didn't see in the attack side of things is, for instance, a group of cycling cells. But I don't have too much time to talk about why this is today. But with this correspondence here, we can actually also integrate the two data sets together by means of projecting into a joint subspace, and then looking for nearest neighbors in this joint subspace, and then matching basically the labels across the two modalities. And actually the cell types match really, really nicely. And we also validated this using the multi-ohm assay. Once we have this matching, because we have a lot more cells here in the single ohm data, we processed further with the single ohm data set. We could actually assign potential regulatory elements to the genes by means of just correlating the attack, so the chromatin accessibility towards the gene expression of nearby genes. So what you see in this heat map here is now pairs of putative enhancers and genes that vary across all the different cell types that we identified. And when we looked closer at genes, that how many enhancers were linked to each gene and how well the correspondence was between the attack signal and the gene expression signal, we found a group of genes that were high, where the correspondence was very high in terms of the correlation, as well as the number of enhancers that were linked to it. And we named those genes genes with predictive chromatin. And interestingly, those genes were highly enriched for transcription factors with known roles in their developmental trajectories. So speaking of developmental trajectories, we also looked at neuronal differentiation specifically. So using RNA velocity, we computed pseudotime values and this trajectory, which was rooted here in the group of cycling radial glia cells, and then branched out towards the differentiating neurons. We projected those pseudotime values into the attack world, and then looked again for matches of potential regulatory elements towards genes that are dynamic along this trajectory. And just a couple of examples here, we see genes that are highly specific for earlier radial glia cells here, whereas we see genes that are more responsible for neurogenesis down here. And when we look at the chromatin accessibility, the matched enhancer elements, we specifically see enrichment of transcription factor binding motifs that characterize again here early neurogenesis towards late neurogenesis. We were also interested in finding whether there might be cooperativity between different transcription factor motifs. So on the one hand, we just computed the correlation of accessibility in the binding sites of pairwise motifs. And on the other hand, we also computed a score which is called a synergy score, which is what you see here in this lower triangular matrix. And this score basically quantifies the excess variability that you see in peaks that basically harbor two transcription factor binding sites from two different transcription factors versus peaks that harbor only one of the two binding sites. So basically gives you an idea of how much of the variation in accessibility across pseudotime is explained by those sites where both transcription factors can potentially bind. And what you see is when you cluster the motifs based on their synergy, you see one group that is interacting with one another, you see a second group that is strongly interacting with one another, and you see one group of transcription factors that doesn't like to play well with the others. And when we looked at when in pseudotime the corresponding genes were expressed that correspond to those transcription factors, we saw that the first group, the interacting group, occurred early. The highly interacting group occurred right at the onset of neurogenesis, and then we have this last weakly interacting group, which drives home neurogenesis later on in the trajectory. And then lastly, because we also were highly interested in disease context and how this could be applied, we also learned deep neural network models that were actually able to predict chromatin accessibility based on DNA sequence. And we then used those models in order to predict what the change in chromatin accessibility would be if we mutated the sequence. And we took basically a catalog of mutations that were associated with autism spectrum disorders in sibling pairs, and specifically introduced those mutations into the sequence and then predicted change in chromatin accessibility. And we found that basically the mutations that happened in the cases of autism spectrum disorder versus the controls led to larger changes in the predicted chromatin accessibility. And of course, many of those mutations also occurred in potential binding sites for transcription factors. So here's one example, for instance, of a mutation in the NFIA motif that also occurs in the NFI gene. Nearby there, we see high dynamics in our chromatin accessibility pattern, specifically with high accessibility in the neurons, in the excitatory neurons. And this NFI gene also has been previously implicated in autism-related diseases. So we would like to follow up further on this, of course, with some more in vitro models, which is sort of an outline for the future. And with that, I would like to thank my collaborators, Alex Trevino, Femina Anderson, and Lakshman Sundaram, and of course also Sergio and Bill. And I would like to thank you for your attention and happy to discuss and take any questions.
Thank you very much, Fabian, for a great talk. You also stayed perfectly on time. It's now perfect time for questions. Do we have questions from the audience? So while I let you think about your questions, I would like to ask you one question. Wait, I wrote it down while you were talking and I need to look for it. Yeah, I was wondering, how did you do the analysis when you tried to look for the correlation of the chromatin accessibility to the gene expression of the nearby gene? So what you did is that you had the attack and you had the RNA. I guess you did that for the paired data, and you were trying to see what genes correlate to what positions in the genome. And with that, you said that you found a list of putative enhancers. And what I was wondering is, did you really look genome-wide? Did you look at a certain area around every gene? Did you have a preset of putative enhancer regions that you tried to validate? How did you do it?
Yeah, so we looked at basically a very large window around each gene and looked for which peaks were around there. And then we correlated the accessibility with the expression of the gene that's nearby.
Okay.
So
yeah, kind of like a Cicero, but with instead of accessibility expression.
Exactly, yeah.
Okay, okay. Do we have questions? Because I have another one, but I don't want to, take the stage if there are other questions. So, okay, then I should. So my question is the following. I thought it was super interesting that you found that there were some so how did it happen when you found a group of genes that had a high correlation with the enhancers in the gene expression? So where the genes, the chromatin was very predictive of the genes, you gave them a given name, I think genes with predictive chromatin or something like that. And then you saw that these were highly enriched with transcription factors, which were important for that particular stage of differentiation. So what I wonder is it that these genes for some reason are indeed more regulated through chromatin, or is it that if you were to look at any stage of differentiation in other cell types, in other important, where in other cell types where other important genes would have been then picked up as the ones which are highly chromatin regulated, and there would be different genes, but these are always the relevant genes for that given differentiation process. Do you see more or less what I mean?
So we thought about this, and we thought that those genes might occur in certain gene modules, right? So we looked specifically also for the overlap with super enhancers, and we actually found some. So these genes are actually seem to be co-regulated to a certain extent, at least, because they also have a high number of putative enhancers into them.
Yeah, I think that is super cool. It's very, very nice. Good. So if there are no more questions, then I think it's time for a break. And I think we can just do a short break, isn't it? We could, for example, meet again at 4:45, like it was planned for the last talk and the discussion of today. And please stay for the last talk. Please stay for the discussion. It's going to be really very interesting. And I would like to thank the speakers of this session. Thank you very much for your amazing talks. See you in around 5 minutes.